بسم الله الرحمن الرحيم. أيها الإخوة والأخوات الأعزاء حاضري هذه الدورة، سلام من الله وبارك طيب عليكم ورحمته تعالى وبركاته، وأسأل الله مساءكم بكل خير. إنه من دواعي سروري أن ألتقيكم في هذا المساء المبارك لنتدارس معا موضوعا هاما، بل أحسبه من أهم المواضيع في مجال المختبرات الطبية خاصة عندنا في السودان، ألا وهو ضبط جودة الخدمات المعملية أو ضبط الجودة في المختبرات الطبية. Quality control in health laboratories. The importance of this matter is drawn from that no one can guarantee the quality and the reliability of laboratory services without applying or setting a program for quality control and applying exactly and effectively that program. Before starting to discuss this matter together, I'd like to display in front of you some terms which are common in this field and which you have to get familiar with them, so that when you encounter them from time to time, you can understand them and you can understand their meanings. Quality, which means providing the services or the qualities as needed by requirements. Quality assurance, to make reliable that every service that have been developed through a given system which you see on place will meet the requirements and the standards. Quality management means controlling and managing and grasping and holding the quality materials and elements that can which they can achieve the quality. Quality management system is a complex and complementary system comprised of multiple elements which are applied together to parallel at the same time, so as to achieve the goals. Quality control is the issue that we are going to discuss, and it is an important element of the quality management. Control materials and calibrators are materials and requirements that you analyze together so as to set the zeros for the management system, for the measuring system, and those are calibrators and controls which you analyze parallel with the patient samples so as to assure the quality and the reliability of laboratory results. ISO is an international organization concerned with the standards, and this material which you are going to discuss is actually initially set and developed by the International Organization of Standards, which is the ISO. WHO, the World Health Organization, is another international standards organization, and it does have a wide and indispensable role in setting this material which we are going to discuss. CLSI is an American institute concerned with setting standards and requirements for the clinical sector together with the laboratory standards, and also it does have a positive role in setting this matter which we are going to discuss together. JCI is another international organization concerned with setting standards for healthcare facilities internationally, as the CAP, which also does have the same role, and standards are those things or phenomena or materials upon which people are agreeing upon and adopted and achieved and actually assured for the reliability and qualification of setting and achieving the quality services or products. SOPs are the procedures and those setting steps by steps procedure adopted to achieving standards. And accreditation is the action of the recognition of different facilities as achieving quality materials and quality standards internationally. If I have successfully presented this material in front of you, and if you are also successful in understanding what I am going to discuss with you, it will be possible and capable. Every one of you will be capable at the end to define quality control and to describe its reliability or its relationship with the overall quality management system. As we said before, it is an important element within the quality management system. And also everyone will be able to describe differences between quantitative, qualitative, and semi-quantitative services that are displayed by health laboratory to the different clients. And that is because quality of laboratory services is actually divided into one of those three categories, either qualitative services or quantitative services or semi-quantitative services. And every category of those quality and of those category of services should have its specific procedures to assure their quality or to control their quality. This material have been driven, as I said before, from a document set by ISO, which is the ISO 1589, but it has been amended by CLSI and WHO in this form of training. And it is then clear that this element, which is in the center, which is the process control, includes the quality control that we are going to discuss. So quality control lies within this system of the laboratory quality management system. It lies under this topic, which is the process control. Quality control, QC, is a part of quality management system of quality management, as we said before, and it focuses in achieving and fulfilling the requirements. This is the definition developed by ISO 9000. Quality control in health laboratories is actually the act of examining control materials of known substances together with patient samples side by side, so as to control and to monitor accuracy and precision, which are the criteria of good and quality laboratory services. And this should take place before issuing or before finalizing the laboratory services and issuing the reports. The purpose of carrying out and adopting quality control programs is to correct the problems, to discover the problems and the weaknesses and the errors, and to solve them before issuing and before providing the laboratory services, so that no health service, no laboratory service will be provided to the clients before assuring or before being certain with the quality of that service. So quality control should take place before reporting the results of the patients. If it happens after issuing the results, this will become another story. Actually, it will not be quality control. It is very necessary for quality control, all the quality control procedures and operations, to take place before issuing the results of the patients. Due to the laboratory complexity, or due to the complexity of laboratory practices, in specific, and generally due to the complexity of health systems, laboratory practices are divided into phases, which are pre-analytical phase and analytical phase and post-analytical phase. And it has been assigned that the errors that used to take place from time to time within the pre-analytical phase are 46 to 68% of the errors that take place, which is actually the bulk of the errors that take place. So it is very important to pay a special consideration in controlling those processes which lie in this phase of quality in health laboratories. And the errors that take place within the analytical phase of quality in health laboratories are 8 to 17% of the errors, actually. This is because those who are practicing in this area are highly professional, and they are the graduates, and they are those who are specialized in the laboratories, and also they use the different technologies and equipment. Some of those equipment are highly skillful and highly clever to work by themselves to control and to achieve the services. This is why the errors in this level are reduced. The errors that take place in the pre-analytical phase of the laboratory quality control of the laboratory quality services is 18 to 47%, 18 to 47%, because In this phase, some people which are not actually specialized in this level, in this profession as laboratory specialists, are not are working in this area, and also it is the interaction area and the interface area that join between the specialists and non-professionalists together with other people from outside. This is why the errors are very common in this area. So when dealing with this issue of quality control, we need to expect this number and this amount of errors, and to take in consideration a specific effort, to pay a specific effort in dealing, in discovering and solving problems in each one of those areas. As we said before, the bulk of the errors in the laboratories lie in the pre-analytical phase, and those are some categories and examples of and causes of why are the errors are centralized in this area. That is due to patient identification and patient preparation, because we need to prepare patients from time to time from different laboratory, from different services, laboratory services, and selecting the site of collecting the sample, and also site preparation of the site from which sample will be collected, and then the tourniquet application and time. Sometimes people fail to find and to palpitate veins for collecting blood, and this results in that the tourniquet will remain fixed on the arm of the patient for a long time, and then the concentration of the blood will alter. So such sample which will be collected by prolonged tourniquet application would not be representative and would not be eligible for examination, and the results which will be obtained from such samples will not be reliable. Proper vein puncture techniques. Vein puncture should consider that the sample collected through this technique should be also representative to the whole blood that is running in the blood stream of the patient. Order of drawing. When it comes to that, you need to collect big amount of blood for different categories of analysis, and if you are going to collect the blood in different containers, it is very important to put in order which container should be used before the others in collecting blood, and that is because for the sake of achievement of quality of the results that you are going to issue at the end. Proper tube mixing after collecting blood in the sample container, which are containing some chemicals for preventing the blood from clotting. It is very necessary to understand the mechanism and the procedure unique and capable for mixing the blood with that chemical so as to prevent the blood from clotting, not shaking vigorously so as to this may lead to rupture or destruction of some blood cells. Proper tube mixing prior of sucking the blood for the analysis, and this is very necessary for the homogenization of blood, so that the amount which will be subject to the analysis will be representative of the overall sample which has been collected from the patients. Correct sample volume is very necessary to have the right volume necessary for analysis. If we take smaller amount which is not satisfactory for analysis, we'll never wait for correct results from unsatisfactory samples. Proper tubes, proper tube handling and specimen processing. This is due to this is necessary for maintaining the integrity of the samples so as to remain eligible for analysis, to be reliable for analysis. Serum samples. It is very necessary to understand what are the serum samples and how to handle them uniquely, how to handle them in the right manner so as to keep them eligible for analysis and suitable for analysis. It is the same story also with the plasma samples and centrifugation of the sample. This centrifugation should be controlled, and everyone using this machine should be aware with the theory with which this machine used to work. Otherwise, he will lose or he will spoil these different samples subject to centrifugation and render them unsuitable for analysis without the understanding that those people will analyze unfit samples for analysis. And specifically, finally, the results will be unreliable. Special handling of blood, handling of blood samples, like those samples which have been collected for glucose, for example, is very necessary from the beginning for such sample to be collected in fluoride isolate, for example, and that is the specific container containing very specific chemical which will maintain the concentration of the glucose and prevent them from being reduced through a mechanism of glycolysis. This is only an example. Similar sample also for different circumstances should be handled specifically and uniquely so as to maintain them and to maintain the integrity and suitability for the analysis. The stability for whole blood and serum and plasma, it is very necessary to examine them at the right time, otherwise to preserve them uniquely and specifically through a mean and a suitable mean which will keep them eligible for analysis and which will save the analytes that you are going to examine and to test and measure in the samples. Otherwise, these amounts of the analytes will reduce and render the sample unsuitable for analysis. Quantitative examinations are, as we said before, this is one category of laboratory services. Through this category, quantitative examinations, it measures the quantity of a particular substances in the sample. Those substances present in the samples, we measure and count them accurately, actually. And this measurement should be both accurate and precise. Accurate, closer as much as possible to the true value, and having the same accepted repeatability. That's to say, whenever you reanalyze that sample, you are going to obtain the same value or a value with acceptable variation from value to another. Qualitative examination methods. These examinations that do not have numerical values, not quantitative values, not quantitative examinations, such as growth or no growth of microbes in cell culture or in microbial cultures, and such as those tests which are reported as being positive or negative, and other tests or similar tests which are reported as being reactive and non-reactive, and those tests which are characterized by color change or changing color after the reactions. These are descriptive examinations and with no numerical values. And semi-quantitative measurements of laboratory services, results are expressed as an estimate of the measured substance, an estimate, not accurate enough, not exact enough, and also not descriptive as the previous category of laboratory services. This is such as trace amount, sometimes trace amount, such as that report which we indicate when we are examining blood for reducing substances, for example, or moderate amount, as we used to report sometimes when we count cells in samples like feces, or when using plus notation, 1 plus, 2 plus, or 3 plus. Also, such as number of cells per microscopic field, when we estimate in the microscopic field. This is just an estimate, although we may specify some values or some numbers, but those numbers are just an estimate. They are not accurate enough. The other or the last meaning or example for The semi-quantitative analysis report are the teachers and dilutions in serological tests, as those when we say 1 over 20, 1 over 40, 1 over 60, those reports which we need to report when we carry out serological tests like Vidal test. Vidal test which we routinely use to detect antibodies against Salmonella or Brucella. For application of quality control, it is very necessary from the beginning to develop a program. It is very necessary to set and adopt a program, and this program should be followed step by step so as to achieve quality. And application of program should begin with setting policies, quality policies from the beginning. From those policies, we need to develop procedures, standard procedures and practices. Those policies and practices should include corrective actions, because the mission of the quality control practices, quality control procedures, is to discover and to point out the different errors and mistakes, and to set system for correction of those mistakes. So it is very necessary to include within the policy those practices and those views of corrective actions, which are very necessary and which are the fruit of applying quality control programs in the laboratories. After setting policies and procedures, it is very necessary to train all the staff of laboratories on the application of quality control system in laboratories. That is very necessary actually. And then through application of that system, it is very necessary to maintain and assure development of records and documentations. By applying quality control measures, multiple records will be developed, and actually utilization of different and getting use of different documents is very common. Is very common as policies and procedures and standard procedures and those materials. It is very necessary to know how to manage information contained and included on those records and on those documents. Finally, we need to review from time to time the quality control data. As we said before, everything and operation that have been carried out should be documented, should be recorded. Those records should be maintained and managed ideally, and subject for revision from time to time to assess the overall system that we have adopted regarding the quality control and detection of the mistakes in the health laboratories. Summary. To summarize what we have discussed, it's very important. Quality control is an important part of quality management system. The goal of applying quality control system is to detect errors and solving them before issuing and reporting the laboratory the patient's results. Different methods actually exist for different categories of laboratory services, and we have discussed this before. Thank you very much for attending.